Start. So hello everyone, it's my pleasure to give a presentation on this topic. And it is multi-omics integration via a network of visual analytics. So yes, Joanne introduced me, so let me briefly just tell something about myself. So I actually have from a medical background, and I did more clinical training from Peking University. Then I did my actual master and PhD, both at the University of Alberta in immunology and metabolomics. Then I did my postdoc at UBC doing more next generation sequencing. And then I went to McGill, and start doing my research as PI, and you can see actually my background is very, very diverse. And my current research area is, I don't know how to define it actually. We do a lot of metabolomics. We both doing wet lab and analyticals. We also do computational development. And we also do a lot of transcriptomics and microbiome, which is my current research interest. And so that's really comes to multi-omics, because we actually deal with a lot of omics, and naturally multi-omics is easier to say, to pronounce, and instead of just try to explain one by one. So I also have a lot of the areas of collaborations and research grant from infectious disease and microbiome studies, also exposed omics, and more recently I have some nutrigenomics studies. So it's really I think nowadays is once we doing omics and big data, and it seems you have to collaborate with a lot of people, and it's just a natural progress. And today my topic will be actually not totally focused on OmicsNet 2.0. It's more about a general concept of how to do data analysis interpretation, which naturally will come to an OmicsNet, because OmicsNet actually is represent a lot of concepts put together, and also why we're doing what we are currently doing. So this is, I think, quite relevant for understanding of kind of get a background history. And the other one beside OmicsNet is a companion website called Omics Analyst, which published last year, not this year, but it's really supposed to work complementary with OmicsNet. So I feel if I want to give a comprehensive picture, I need to present both, and hopefully you feel it actually more clear when you compare each other. And lastly is a future perspective, because this area is actually moving very fast, and how we're going to proceed and to the next stage, which is I'm asking myself all the time. Also, it's really try to figure out what's the most promising strategy, move on. So this is a very interesting topic, and I hope someone will ask me very good questions. So I really want to learn and see what other people think about. So before we move to multi-omics, and here's my perception of general omics. So omics is all about high throughput molecular profiling using a variety of technologies, high throughput, and hopefully it's low cost. Yeah. So there's a lot of technologies, and so far, from my own experience, most commonly used is sequencing. So next generation sequencing is dominant. And I'm doing metabolomics, so mass spec is also widely used. I guess probably NGS is, let's see, 17%, and MS is 10%. Then there's imaging, there's probably other technologies which I'm not quite familiar. But overall, as I feel, if we focus on the NGS and MS, and we can cover almost majority of the large data, which is publicly available. And from this technology, we get a lot of omics data, and we really want mapping out different layers of our whole system. Like on the right-hand side, we have metabolome, proteome, transcriptome, epigenome, genome, and we're using that. We really want to do diagnosis, understand the disease. So everybody is kind of on the same path these days, and really want to see which one working best. But I guess most of us, you don't want to learn programming and do from the very raw data processing and get your table and do statistical analysis, generate figures, which is very specialized thing, and it takes a lot of time and takes years of experience to training to get there. So here it's really coming to how we do bioinformatics to help majority of the researchers to do their job, to focus on understanding disease, understanding biology rather than writing their code. So this part is actually why I'm continuous just talking about this, is because I'm from biomedical background, and I spent 10 years of training, almost get more than 10 years all the way to the current. I don't think people should really switch their career and do that. Overall, that bioinformatics or computing in general is becoming easier, but still doing well is more take much longer training. And the tools supposed to be easy to use, and most people should be able to get their job done just using tools by clicking rather than writing their own program. And with this philosophy, we actually have the kind of patterns. How do we do data analysis, and how do we develop easy to use tools? And now I'm just cover what is the main patterns to do omics data analysis. So it's mainly about 4 stages, and the data processing and the quality control, which is most critical, which also is related to what type of data you're getting. So sequencing and mass spec require very different expertise or packages to pre-processing. And also quality control is critical, and you need to have some experience to see what's good quality or what's garbage. So this part is more or less standardized, but it really require a lot of the initial input to understand, to feel comfortable. After we get the raw data processing, we move to the next step, statistical analysis and visualization. And this part, I guess, majority of you will be familiar, and because it is really, we see a lot of publications and colorful figures like box plot, heat map, and some other classification views, biomarkers. And if we get these significant features of biomarkers, and we really want to do is a functional interpretation, and we want to write a story, what's the biology behind this, what's the mechanisms. So here's some of the commonly used tools called enrichment analysis, pathway analysis. So there's definitely other approaches, but this is the main thing. If people want to do this raw data processing stats and functional interpretation. And there's also some other more unique features like causal inference biomarker analysis. There's more. So I'm collaborating with new researchers from more specific field. They really have some special analysis, which I never heard about, but gradually I learned it. It seems quite interesting. And so that's the previous slides I covered is very, very general. And but it's once you have that framework, you go to each individual, became much more natural once you have that. But the other part is if we have this raw data processing, we're going to get a huge table, and you usually don't open it using an Excel spreadsheet. And if we want to analyze it, you also don't want to analyzing it as a whole. All of them will treat it equally. And because omics data really contains a lot of noises, And if you use all of them, and it is computationally intense, and also contains a lot of the false positives and potential overfitting. So data need to be filtered and needs to be distilled. And the next one I would like to emphasize, which is also core for this presentation, is we need to couple stats with visualization. Why is that? The current statistics, disregarding how advanced, they cannot capture data, or even they can, we don't, our brain cannot follow, okay? So a lot of people just use p-values and talking about p-value less than 0.5 and think that's it. That's definitely not the good way. And nowadays we really talk about, we use statistics cautiously, and statistics is helpful. And we also need to use our own judgment based on background knowledge. So visualization is critical. And in addition to visualization, thinking, and we also need to use domain knowledge, which is called context. So you need to understand literature, what's going on. And is that current finding is fit with the literature or contradict. So overall, that data analysis whole package is not just one method or one particular visualization will be it. So it's you really need to understand the big picture. So whole idea is from raw data, process data to informative data. Informative data means data really, you can think on it, you can make decisions on it. So there's this part is hard. Why? Because informative to me is not informative to you. We have different backgrounds. So it is hard in this case, but overall that if we are talking about researchers in particular area, we actually can be more homogeneous. We can provide more context to help people understand their data. For example, we talk about omics data, we do have the metadata describing the data, patient information, the age, and the disease or whatever the other types. And we actually know other relevant information, like if this is a particular prostate cancer, and we can actually get other published prostate cancer information, knowledge on that particular disease. And overall, we have more general information like pathways, networks. So overall that we can link from the current data to a bigger picture. And from this big picture, we can understand better of what your data, whether it's consistent or novel, or there's something that's unique. And so this is part is how we use context to help to understand the current data. And I put a sentence here is that I don't know who's going to ask. Overall, that my current thinking is that all of our data integration mainly is driven by our own ideas, our own thinking, our own understanding. And we do use visualization, we do use statistics. And definitely we do hope in the future this artificial general intelligence can do more help, but at the moment, this is not there yet. So in this context, how we're going to understand the data is by providing better stats, better visualization, and better context to engage and empower the researchers, the domain expert. So we need to provide we need to digest the big data to be more kind of digestible. And we need to bring a big background to be more interpretable, such as pathways, biomarkers, or something else. So in overall, so we put everything accessible, and so people can understand the data easily without writing their code or think about their bugs or stuff. And here is that we talk about visual analytics. And if you are from computing science, visual analytics have a specific term. So what it should be. I come to visual analytics just from my own understanding. So I think it's a visual and analytics. So you need to integrate your visualization with statistics or machine learning. So when it combined together, it's visual analytics. And one thing is that I put here is a lot of the disruptive discoveries are often driven by hunches or leap of faith of researchers by interacting with data, which is actually a lot of researchers, including me, I like to stare at the data, see a good visualization and think about it. So I really enjoy spending time with the data, but not just stare at the Excel spreadsheet and actually at a good visual representation. So the other part is currently deep neural networks, a lot of people are doing it, but mainly still a black box. So I cannot think too much rather than just try to believe it. A lot of time I just cannot give up because I really want to understand what's going on under the hood. So deep neural network is, I really want to hear experts' opinion, whoever doing it a lot, and give me more insight. So far that I'm reading a lot, it's just still there's back and forth with how to integrate better with the current research framework. So on this right-hand side, I put some good network-based visualization, which on the top is a 2D and on the below is 3D. And both is quite informative. I can spend a lot of time thinking and interacting and feel it. So this is a kind of visual analytics for network, which we have been implemented. And I think it's informative when you present it in the right context. So that was a concept, and here we need to implement it. Just because we talk about it, and it doesn't really matter until you can do it, or you can let other people do it and show it works and working well. And how to do that is we're using several technologies, and one is we do have a local cluster. This is for our raw data processing. This is mostly not open to public, just because it is quite expensive, and raw data uploading hundreds of gig of data is quite hard to move in from remote places. So mostly data generated locally from my stack. But from here is that everybody can use it. One is using cloud. So if you use a table analyst, network analyst, you will access it using Google Cloud. And the other part is your own computing power is the browser. And most of us have very powerful browser, and almost all the visualization is through your browser. And last part is your brain. So we're talking about, we have 4 different computing engines. We have a cluster, we have cloud, we have browser, we have your brain. Actually, most time with computing happens is actually in your brain, because when you start interacting with data and it start thinking, and actually the cloud is not working hard. I can see we can have thousands of users, and the cloud is not actually that going crazy. And just because when you visualize your data, which is on your computer, and your brain is thinking, and this is most important part. And what we are thinking in the future is called edge computing. And this is more about how we leverage other resources, devices to make the computing more affordable and faster. Because so far that if we just using a cloud, and at some time, especially we're talking about deep neural networks, it still costs a lot of computing and time. So we need to think about the better ways to make it more affordable. So that was the framework, and we actually also doing a lot of the based on that, we're doing kind of raw data processing. We're developing a lot of raw data processing algorithms. We make it, even raw data, we can make it 100 times faster. So top is the high resolution mass spec, and below is NGS. We able to just optimize the algorithm, make it so much faster. So even if it's run a cluster, it will be finishing in a few hours rather than several days or weeks. And here, once we get a table, we can run on a cloud, which is almost as instant. So you upload data, you click, you'll get a result. So this one is more or less okay. And what we see in the future is we think we are going to use more AI-enabled technology. We also think that VR or augmented reality or mixed reality will be more accessible in future. So overall, that we think we will be able to make the data more affordable and data analysis more enjoyable, and we can do it. And here is the tools, and there's actually a lot more tools below, but here is the tools we developed over the past few years. And the top is the multi-omics analyst. You can see we have almost 500,000 users. We continue release almost every three years. We also publish tutorials. And we have network analyst. Network analyst is for RNA-seq, but RNA-seq actually we moved to another tool called Express Analyst. So you have no problem to do network analyst, but when you do a transcriptomics, you're going to do another tool. So why is because we think expression or transcriptomics need to be more dedicated for a single cell especially. So we want to remove it. This one focus more network. And we also have microbiome analyst. So this is also used heavily. So if you do microbiome study, you should use this. And we have other tools. So the last two tools, OmicsNet is Omics Analyst, relatively new. You can see the users not so many, but it's one of a kind. So it's have a very good concept, also good interface. So we just really want to see people start using it, give us feedback. So we will continue to improve it. So this is a multi-omics analyst. We've been just developing these tools for almost 12 years. So it's being used by a lot of people. And we can see it's continue to grow every year. And we have about 2,000 or 3,000 users every day. So, and this is one of the, we can see the earliest tools, but even with this, we try to generate a very beautiful visualization. So most of them is interactive. So you can get this classification and biomarkers and pathways, and you can have a very end is PDF report. So it's really present in a way that it's intuitive, and you see what's the most features and what's the patterns there. So this is more new features. We allow people to do their raw picks. Basically, this is the picks is from mass spec, and for most people, it's not interpretable. And if you direct analyzing it, you have no feelings about what it is. For example, you can search a genes and metabolize, but picks is almost uninterpretable. But picks you can direct generate from mass spec. So we use a GSEA, for example. We allow people to directly interpret their metabolomics data from picks to pathways. So you can see it's much more interpretable, and all these kind of things is pathways. We can allow people to find the patterns directly from pathways. From the patterns, you select this patterns, you do clustering, select patterns, and from patterns, you can do pathway analysis. So this is a core concept of visual analytics. This is not a network, it's a heat map visual analytics. So here's a network, here's a heat map. You cluster and find the patterns, and you use your mouse to select patterns, and you perform analysis to see what's going on with these patterns. So overall, that really allow you to think, react, and understand. So this is a key concept of doing visual analytics. Now, I'm basically ends my introduction about individual omics, and how do we now we gradually move into multi-omics. So what should we expect from multi-omics? And so So the thinking here is that multi-omics is very different from system biology. Okay, system biology really need to aiming to understanding mechanistics and including everything, how it works together. So it's almost we talk about this knowledge. And if we talk about individual molecule like protein 3D structure, we really think that's a very deep interaction, mechanistic stuff. And multi-omics are right in the middle. It's give you the patterns and the trends. And from here, you generate a hypothesis. And from hypothesis, you can really go back to mechanisms or back to more simulations. So, so how do we do the data analytics is really the focus on patterns and the trends. So how do we get that visualization? Stats or machine learning became very critical. So how do we do multi-omics data integration? And we see there's three main approaches. One is user-driven, and this is really reflecting what I just mentioned. Currently, most data knowledge discovery or integration is user-driven, and it happens in our mind. And we just read and think, and back and forth, we write the papers, and that's user-driven. But tools is coming and allow people to perform more automated integration. For example, knowledge-driven. This is OmicsNet. The other one is data model-driven. So we do have a mathematical model about how things should work and feed the data there and update the parameters. So this is all of them is valid, and I'm going to discuss them in the coming slides. So user-driven we have a good understanding of our data and a good understanding of the biology behind it, and we do some regular single omics analysis, and we incrementally refine and developing the stories or narratives. And we also read the literature and try to understand what's going on. And finally, we want to put it together. And what's the final how to say story come to your mind? This is really, you are questioning yourself and back and forth with the data and with the literature. And this is quite natural, but it just takes very long. And good data, it takes months or if not years to get there. So I see when people ask me about are they tutorial for beginners for our tools? Yes, it's designed to be very user-friendly. You can click to use, and we also have a lot of tutorials. You just need to go to the website and read and find out. So we really want to make it easy to use. And that's the user-driven. And that's natural, but it takes time. And knowledge-driven will be faster. So knowledge-driven is that we need to first have an extensive knowledge base, continue relationships among different molecular types. So for example, pathways, regulation networks, and other common sets define disease or phenotypes. So all of this is helpful knowledge that will help us to find the patterns and associate the patterns with some functions or pathways. For example, here, this is one of the knowledge database that contains a lot of the information from EMBL, from NCBI, from regular, and what's the transcription factor, what gene ontologies. And so all of the network pathways will be there. And then you continue perform your individual analysis and you identify significant features. Then can you map in this all the features to the pathways networks and you can visualize them, see what the patterns that collectively all the individual molecules on that big knowledge framework, what it tell you. So this is easy to see, but doing it well is not so easy. It's just because you need to have a lot of knowledge networks there, and you need to visualize them, and you need to analyze it on top of that. So this is exactly OmicsNet we try to create, and how it to help people doing that. So let me repeat what for knowledge-driven is that we do an individual omics analysis. We identify signaling features or signatures from them. Then we project them to this knowledge framework. Then we try to identify the patterns from this knowledge framework. How do we do that? Is we can do a simple enrichment analysis on that, and we can do graph-based mining, like a price collection networks, stay in the forest, PCSF, and there's definitely multiple others to do it. So overall, that once we have the signatures, once we have the project signature to the knowledge framework, we can have so many tools to help you to analyze, visualize, and back and forth, you will develop a lot of theories behind it. So overall, that the key part actually is 3 and 4. How you allow people to back and forth interrogating the data to understand. So that was the OmicsNet. I'm going to talk about it more, but here's let me talking more on the data-driven or model-driven. So data-driven, model-driven is more recent and also active research area. Why? Is because it is really feels nice about you have a model and you can do more mathematical test on that, and the result more clean, how to say, more already have a conclusion built in, how to say, let's see that. The thing here is that it is really dense, and the result is very multivariate, and it's not interpretable for majority of researchers. So most commonly used is dimensionality reduction. So you can think about PCA, principal component analysis, but when you apply it to multi-omics, and it's much, you can see 10 times more complex than PCA. So this is a very, how to say, very dense mathematics algorithm, but also very promising. So I guess that with more data, with a better, how to say, education, some of the tools and will become more popular. We probably understand better how it works, how to interpret it. So this is the part is currently working here. So overall, that my idea or what we have been doing in the past few years is really quite try to allow people to do single omics easily and doing multi-omics easily. And we have developed OmicsNet, which is network-based, knowledge-driven visual analytics. So this is the one I just mentioned, is knowledge-driven. And you have your signatures, you project to this network, and you visualize and perform analysis on these patterns. And omics analyst is the machine learning, multivariate statistics. And you can perform a lot of the commonly used dimensionality reduction on top, and interactive visualize the pattern. So overall, that result is presented in a way that as best as possible, but the math behind it is still quite hard. Why? Is because we don't usually see this mathematics. We see t-test, we see ANOVA, we see PCA, but this one is 10 times more. And there's a lot of gaps. If I just see this word, you don't get it. So overall, that I do see in the coming years, it will be easier, but there's a lot of effort in both communities. So now I'm touching OmicsNet. So OmicsNet is for network-based knowledge presentation and visual explanation. And I guess most of you have used the Cytoscape. So Cytoscape was also quite popular about 10 years ago when I did my postdoc. But Cytoscape was a desktop at the time. And I really like using web-based. So network analyst was developed earlier, actually first one to do web-based network visualization. And it was working very well, actually faster than install Cytoscape locally. So we were very proud at that time. So gradually, we found a 2D network is okay, but 3D could be very cool because 3-dimensional networks, you can see something you don't usually see. So a few years ago, we used WebGL, which is people use for developing games. And we use to develop a network visualization. So you don't need to install anything. You can use your browser and visualize the network in 3D. It's very cool. And you can see the this top network, you can see like that. And you definitely can zoom in and out or rotate. And you see all this coloring halo there is actually just a seed, which you have your proteins, you want to who's interacting with that. So it was very satisfying, just very looks beautiful, but how you actually get more from that is not quite clear. We can also actually do this in a VR. We can actually, you put on your VR goggle, you can see it there too. So that's one of our hidden goals. If a goggle is really just like iPhones, everybody have it. And we actually can move it to the virtual reality easily. So on the bottom is that we just add in how do we reduce the complexity from the 3D network to a more digestible format. Is we add in bubbles. The bubble means that we put them into a different modules and color them differently. You can actually interact them separately. It's make it easier. So overall, that we really try to explore how we get a lot of the data into a network and how we can explore, visualize network in a way that is not overwhelming and actually making sense. So that was the OmicsNet visualization. We developed using latest technology, WebGL, make sure we can view it in 2D and 3D. And actual VR, if you have the goggle. And this is OmicsNet, and it accepts a lot of the signatures, basically signatures, your significant molecules from your study, individual omics, like SNPs and microbes, LCMS peaks, and genes, proteins, transcription factors, microRNAs, metabolites. So you can click to upload and try to connect with your other omics or connect with the knowledge, basically other key molecules. So, we have a lot of the well-known molecule like microRNA interact with genes and transcription factors and protein-protein interactions and metabolites protein enzyme interaction. So we have a lot of understanding, and we can actually also link to SNPs to potential genes and peaks to metabolites and microtaxa to metabolites too. So there's a lot of things we can associate things together, like reactions, regulations, associations. Once we have things together and link that, we can start think, we can start to come up with possible hypothesis. So yes, this is one of the output. If you go upload a list of genes and you want to find out what this gene means with a big context, and the genes can be created really just if you have a lot of genes, you want to see who's interacting with who within your genes. That's okay. It's basically you're talking about zero order networks. Zero order means you don't introduce, you just interact with yourself, zero order. First order is that you connect with a friend. So basically the closest neighbor, and you can also move on. So if you only have one or two and you want to expand, it's okay. But once you have hundreds and you want to expand friends and friends' friends, it can be very large and it could be very overwhelming. Most likely is if you have a large list of genes, you really want to see who's directly interacted with who. It's zero order. First order, if you have like a 20 or 30 and you want to expand, you get more context. And so you can use the first order. So the result, you can visualize it 2D or 3D. And here it just shows this is almost all the seeds as actually densely connected with each other. Why is that? Just because the function is very coherent and they connect with each other naturally. And the story is very, how to say, strong. And you know, you almost don't have other hypotheses. You only have this one just because they are very strong hypothesis or functions there. And here is at multi-omics, you create a network and you want to see how many patterns or modules there. And for example, you can have almost 20 different modules. And among them, this purple one is really strong. And you double click and all the background is fading off. And this one going to the front. And you can see this is some amino acids and genes and microRNAs. So you actually can see, put it into different layout. You see one of the regulators actually interact with the other molecules. So overall, that you can from very complex big network into individual modules, and from module you can find the potential mechanisms. So it is really designed to be serve your needs, serve your queries. And this is more latest features in OmicsNet 2.0. And we added more support for 2D. So this is protein, microbes, metabolites, and as blue one means seeds. We have, so this is also very dense, densely connected network. Internally, they connect a lot. This is a nice thing because that means they have very coherent story. And this 2-dimensional, this is 3-dimensional. We put on different layers. It's automatic. So that means that all of this is each omics have a different layer, and between omics they are linked together. So you probably need to add in these labels. These labels you need to manually do it, but most of other things you can almost generate it automatic. We do have these galleries. And you should see you have all different kind of the arrangement and stuff. So we don't know what type of things you can generate. We just explore a little bit. And this is Omics Analyst. So OmicsNet, I mentioned, it is for knowledge-driven network-based visual analytics. And Omics Analyst is our try to put this data-driven multi-omics integration there. And so you can see we do have very clear pictures in mind when we design this website. We think a network is helpful, especially we just put it on the side of the wall, and the heat map is also helpful, the other side of the wall. And in the center is a rotating 3-dimensional scatter plot, because most of us actually can view a scatter plot easily. And you can actually go into this room and explore your data. And so this is really the vision when we want to develop this. Eventually, so we put all of this is 2D or 3D, it's okay. It's just that you cannot have such a room. Why? It's because you need to have your goggle. And we realize the goggle is, and the browser is not, most of the browser does not supporting that. I use Mac, and the Mac browser don't have a good GPU. So if I'm doing that, the GPU will won't support. So it's so we do have the capacity, but the hardware is an issue at the moment. So what's behind this Omics Analyst? What's the models? Multivariate statistic algorithms. So we actually have a lot. We have almost 12 different algorithms that are commonly used for multi-omics analysis. So I don't want to go to details, but so if you really want to use it, you need to read more to learn more. Overall, that we have the 3-dimensional scatter plot, and you can from the patterns, you can reduce the patterns to not individual data point, just to their to a bubble, because this bubble represents the patterns. Inside each bubble, we have thousands of data points, so you don't want to see them. And we can also see their connections and the different arrangement. For example, this arrangement really clearly shows the molecule in the center play a key roles here. And you can put a lot of things together. You can see that this is single cell. I'm just not sure it's single cell or not. So you can see a lot of the data point and the sample separations and the key biomarkers and all in one stuff. So now is that we have a lot of tools. So we're talking about 2 or 3 tools today, but we actually have about almost 20 tools. And it's became an issue when people ask questions because we don't have time to answer them or through the emails. And now we just created a Omics forum recently. And we ask all users to ask a question through the forum. So when we answer them, other users can see it, can benefit from it. Also, other users can help answer questions if they are more advanced. So this is one. And overall, that we continue to see the users increasing with steadily. And here's some future perspective. What's the future for data analysis? And because we are generating data easily and more going to be generated. And one thing is that I think you don't need to write a program, but you still need to click a button to move on. What's next? What's next is you talk to your tools, it will do analysis. So this one I'm thinking is it's called a chatbot, right? We do use chatbot a lot, and this is my daughter. We attending a conference, and she talking to Siri back and forth and get a lot of things done. I'm really inspired by that. I think voice recognition is more or less easy, and you can really talk and program. You just do ANOVA on my data. They will do it. So when I think about this and just searching online, actually, some people already show the prototype is doable. So yes, just sometimes the computer does not sure what you mean. They will double check to make sure it works. So I think using keyword text or just using a voice to do data analysis is clearly doable. And the one is that can we do it using like more augmented reality or virtual reality, which really I think should happen now, but it seems taking longer. So this is something I was thinking data analysis should make it easier. But all these companies seems they are working hard to get the affordable goggles in future. So if that happens, then our tool will naturally go there because we make it a 3D. The other part is deep neural networks or AIs. So that's at the beginning, I'm just thinking about how AI help with multi-omics. So this one, I'm really waiting for anyone who is doing AI and share the experience, because I'm learning a lot, but I just don't know how we easily can do it. One is computing is so heavy, and you can do it by your own. But when you have large data running for a long time on a huge server, and it's not easy for other people to use. So for me, it's you doing research on it is fine, but when you want to share that approach with other people will be hard. And this is my almost the second to last slide, is overall that disregarding what I'm saying, I do believe everybody is working hard towards a more easy, friendly, or pleasant data analytics in the future. So when we are working together, and also computing will be cheaper, and it should be easier. So my vision is that we have the community, we have the chatbot, and back and forth, we'll understand our data. So this is my last slides, and this is my lab, and we funded by NIH, Genome Canada, Genome Quebec, and also McGill. Thank you all. I see.
Okay, thank you so much for that, Dr. Xia. What a superb presentation, especially with a lot of these good tools, great tools for multi-omics analysis. We just want to read some of the questions, especially from Adam here. He mentioned, yeah. I can, yeah, okay. Adam, would you mind muting probably, or unmuting? You prefer that? Or I can just read.
Yeah, I read that.
Okay,
The thing that
all right.
DNA element or multi-type of IGV, that's fine. The thing that I don't deal with that type of data. So unfortunately, I don't have too much comment on that. If you're doing this this type of analysis is co-localization. I don't know the big context, but overall that I developed tools because I personally involved in that, and I don't involve too much with this DNA elements. I don't know it's epigenomics. I'm not quite sure what's it. The stuff.
Thanks on that.
I don't know the particular question. That's the thing. It's just describe something.
He had another question a little bit above that. Page 22, what does connect through knowledge framework rather than connect at raw omics layers mean? I think probably looking at the manual for OmicsNet.
Oh, let me see. Page 27.
22.
22. This one?
Connect through knowledge network. Yeah, it looks like point 2 maybe jurors from each to a knowledge network instead of raw.
Yeah.
Oh, yeah, okay. So might interpret a little bit. I think your point of doing it with like from the knowledge driven level rather than just looking at the raw data. And you're saying his PI looks at NGS data at the raw level to try to make connections, and maybe we can elaborate a bit more on that.
Yeah, well, I don't know if overall, let's say if we do a large scale omics, we do it from raw level. It's yeah, that's okay. I see the point. If you have a overall framework like you talk about IGV and you have a whole human genome out there and you lay out, that's naturally fine. My context here is that we have a knowledge framework, which is we talk about the millions of nodes, and we want to map in there. We don't interest in all. We only interest in more significant features in that network. So if you are doing the IGV, I think the similarity is that you potentially I don't know, you can do a differential analysis. You only want to map in this significant elements to that genome and see where it's enriched, the peaks enriched in that genome. So you don't want to see something down the change. You map in there, or it's not too much interesting at the stuff. Not sure I understand. Correct.
Okay. And we have a question from Bijou. Feel free to unmute.
Hi, Dr. Xia. It was a nice presentation. So I have a quick tangential question. So, as more and more knowledge is being integrated, I'm sure that these have a background which you're using, like, background knowledge for integration. But is there an effort underway where you are in future, maybe you're going to learn from the data itself while interpreting or digesting the information for the integration?
Can you clarify a little more? I think I get it a little bit, but I just want to see.
Yeah, so when you integrate these data, like different omics data, you're probably basing it on a knowledge that is already existing in the literature, right? So yeah. My question is, like, is there an effort from your side, like from your lab, like would the data itself be providing the information? Would you learn the data directly from the, like whatever you're getting, the raw data, and then trying to derive new knowledge from there before integration?
Yeah, that's very good question. Yeah, I think you learn from data, get more. I think definitely that's attracting. I need to set a framework. You are talking about so many people using the tools and upload their data. Can we learn from that data? Is that?
Yeah.
I think that's on my plan. So if user willing to share the data, and we definitely can learn and continue to learn from that, accumulating and compare with this across the time. So at the moment, all my tools is working by just analyze the data and delete the data. They don't store anything. If we want to store, the whole different framework need to be created, and also user need to consent to doing that. So yes, I agree with you. It's a actually a very good opportunity to do that. Yeah.
Okay, thank you.
So I think we're actually at time going over. So thank you so much. That was a very interesting talk, and looks like there's a lot of very useful tools out there that are kind of broadly accessible to people. So that's wonderful to see. I put the links in the chat if anybody was looking for them too. Yeah, so thank you so much, and thanks everybody for coming.
Okay, thank you.
Thank you all.
Bye-bye.
Have a great day, everybody.